Welcome back to part 3B of factors governing lactic acid formation in skeletal muscle. This video will continue from the first one where I covered why insufficient oxygen in the mitochondria can lead to a greater formation of lactic acid, and how if we have a greater glycolytic flux, i.e. we're relying more on glycolysis during exercise, there's a greater production of pyruvate and NADH. The reason why we might have an accumulation of lactic acid is that during higher intensity exercises, we recruit more type 2 muscle fibers. These are the fast, powerful muscle fibers that allow us to do a lot of work, such as sprint and lift weights. So let's go through a few differences between our fiber types. For those of you who are advanced students of exercise physiology, you'll know that there's subtypes within type 2 and type 1 muscle fibers. But we're just going to go through the very basic differences between the two. Okay, so we often refer to type 2 muscle fibers as fast twitch muscle fibers and type 1 as slow twitch. And like I said, there's subtypes within each of these. And so our recruitment pattern is that during low intensity exercise, we're going to primarily rely on type 1 muscle fibers, but as intensity increases, our motor unit recruitment pattern is such that we're going to recruit more type 2. If we look at the number of mitochondria contained within type 2 versus type 1 is going to have a very high density of mitochondria, while type 2 is going to have a lower density of mitochondria. For the diameter, your type 2 muscle fibers are very large, protein-packed fibers that allow us to do very high force muscle contractions, where your type 1 are much smaller in diameter. In terms of capillary density, so therefore blood flow delivery, there's going to be less capillaries in type 2 versus type 1. The fuel storage, there's much more fat stored in type 1 muscle fibers. So that's what these IMTGs are intramuscular triglycerides. So there's greater fat storage. Whereas in type 2 muscle fibers, you're going to have more phosphocreatine and glycogen stored. And finally, there's a difference in the lactate dehydrogenase isoform. In the type 2 muscle fibers, there's a form of lactate dehydrogenase that readily makes lactic acid, whereas in the type 1 muscle fiber, it more readily converts lactic acid back to pyruvate. So let's go through each of these in detail. As exercise intensity increases, there's going to be a greater recruitment of type 2 muscle fibers. So let's look at what happens when we recruit more type 2 muscle fibers in terms of mitochondrial density. If a muscle fiber has more mitochondria, it's more likely for pyruvate and NADH to enter the mitochondria. If there's more mitochondria, there's more places for pyruvate and NADH to go when it's formed in glycolysis. And therefore, you won't have the problem of a backup of NADH and pyruvate in the cytosol. Remember, if there's a backup and less places for NADH to go, there's more likely for lactic acid to be produced. Secondly, if there's more mitochondria within a muscle cell, there's more aerobic enzymes, and therefore more Krebs cycle enzymes and more proteins from the electron transport chain. This means that there's a better capacity to oxidize the products of glycolysis. Therefore, if we're recruiting more type 2 muscle fibers, there's going to be less mitochondria, and that means that there's going to be not as many places for NADH and pyruvate to go when they're formed in glycolysis, and less enzymes within the mitochondria to deal with these products. If we look at the combination of mitochondrial density, fiber diameter, and capillary density combined, we're going to be talking about something called diffusion distance. So what this means, it's the distance that oxygen has to travel from the cell membrane, i.e. at the capillary, into a mitochondria. And so there's three factors that can really influence. Type 1 muscle fibers have a smaller diameter, have more capillaries, and have more mitochondria, which means there's a shorter distance for oxygen to travel to get into the mitochondria. And as we know, if there's more oxygen in the mitochondria, there's less lactic acid formed because there's the final acceptor of electrons. If we move on to the type of fuels predominantly stored in the different types of muscle fibers, we know that fatty acids are stored in greater concentration in type 1 muscle fibers, while glycogen and phosphocreatine is stored predominantly in type 2 muscle fibers. So if fatty acids are the predominant fuels, pyruvate will not be made. If there's no pyruvate, there's no lactic acid formed. And so if we go back to previous videos in my energy systems pathway, when fatty acids undergo beta oxidation, the product is acetyl-CoA. This totally bypasses the formation of pyruvate. And if you remember, pyruvate is essential for lactic acid formation. So in type 1 muscle fibers, where fatty acids are the predominant fuel, there's going to be no substrate for lactic acid formation. Whereas if we use type 2 muscle fibers, where glucose and glycogen are the predominant fuel, we're going to have an excess formation of pyruvate, which can then go on to form lactic acid. Finally, let's look at the LDH isoform. Recall, this is the enzyme that speeds up or slows down the formation of lactic acid. There's more than one form of lactate dehydrogenase. The way this enzyme is expressed is different between type 2 and type 1 muscle fibers. So recall, lactate dehydrogenase is the enzyme that catalyzes the formation of lactic acid. In type 2, the isoform, often referred to as MLDH, readily forms lactic acid, in that only a little bit of pyruvate and NADH needs to be present to push the reaction towards the formation of lactic acid. In contrast, in type 1 muscle fibers, the isoform, often referred to as HLDH, does not readily form lactic acid. There needs to be a significant amount of pyruvate and NADH to form lactic acid. And actually, in contrast, the lovely thing about type 1 muscle fibers and a few other organs that contain this isoform is that lactic acid can be converted back to pyruvate and NADH. We'll explore that a little bit further in the next video. So as an illustration, when MLDH, the isoform found in type 2 muscle fibers, is present, it only takes a little bit of NADH and pyruvate to have a reaction occur where lactic acid is formed and NAD. In contrast, when you have the HLDH isoform, it takes a lot more substrate for a reaction to happen. And pyruvate and NADH can be in much higher concentration before lactic acid is formed. We've gone through a few differences between type 2 and type 1 muscle fibers. And we know that when more type 2 muscle fibers are recruited, there's a greater production of lactic acid. In this video, we've covered some of the factors governing lactic acid formation, and they include insufficient oxygen in the mitochondria to accept electrons in the electron transport chain, increased reliance on glycolysis and therefore a greater amount of products of glycolysis being formed, and finally, a greater recruitment of type 2 muscle fibers. In the next video, I'll cover the fourth reason why we may have an accumulation of lactate, and that has to do with the clearance of lactate from the blood. I would encourage you to move on to the fourth video in this lactic acid series, where we'll talk about why lactic acid matters for exercise performance.